Hi, I'm Jerome Henagin, and my project is on the effect of monodiamond trihybrid zebrafish crosses have on the phenotype of their progeny. And so zebrafish or Danarerio are part of the tropical freshwater fish and part of the minnow family. They're found in the Ganges region in the eastern Asia. And so the gray wild-type zebrafish have distinct horizontal stripes, and the transgenic organisms contain gene or genes that can be artificially inserted. They can be born with or without stripes and can fluoresce blue, red, yellow, purple, orange, and green. And this is a jellyfish protein that is inserted into the muscle of the fish. And the genotype is the genetic makeup and what determines the phenotype. And the phenotype is how the organism looks and behaves. And so my question is, what effect does the phenotype of the monodiamond trihybrid zebrafish crosses have on the phenotype of the progeny? And my question was, my hypothesis was, if the genotype of the monodiamond trihybrid zebrafish crosses are known, then the phenotype of the progeny can be predicted. And so I did a study at the University of Minnesota Duluth, or UMD, at the Swanson Science Building, and I examined 4 mutant strains, red, blue, green, and purple, and I recorded the phenotype and genotype of the mated zebrafish, and I used 1 mating tank per fish I mated, and I used 1 male and 1 female, and I used 1 plastic aquarium plant in the tank, so I provided a environment that was suited for them. And I placed a compartment with slits in it, as you can see right here, so the eggs, when they were born, go through them, and so the fish couldn't do anything to them. And so I labeled these tanks with the genotype and the gender of the mated fish, the adults, and the date. And after I mated these and put them in the tank, I left them in the care of the qualified scientist, and I came back the next day. And the cool thing about the zebrafish, they can lay eggs within 2 hours, and so the next day I came back, the lights of the fish room was turned on, and the eggs were laid. And after those 2 hours, I collected the eggs, and I put the female and the male adult fish back into their original aqua tank. And so the eggs I collected from the lower tank, and I poured off the excess water carefully, and collected the eggs in a petri dish and brought them upstairs into the fish lab. And I used a microscope to sort the eggs from good and bad, and under a microscope the bad eggs were black and the good eggs were a clear color. And when the progeny were big enough, I determined their sex and their genotype, and I sorted them by the parent cross monodiamond tri. And when I had to wait about 3 months for them to fully develop their color so I could determine their phenotype. And during those 2 to 3 months, they were in the care of the qualified scientists. And so I did a Punnett square and chi-square analysis, and using the Punnett square, this is how I got my expected number of value of progeny. And using the chi-square, I compared the observed and the expected numbers. And so this is my first table of my monohybrid cross, and this is across a blue female and a gray male. And I got a 50-50 of blue and gray. And this is the chi-square of the monohybrid cross, and I looked at the observed and the expected, and you can see that there was over expectancy of gray and under expectancy of blue. And using this chi value, I looked at a chi table, and I got a probability of 20 to 30%. And this is my dihybrid cross. I crossed a red male with a blue female, and I got a 25% for all colors: purple, blue, red, and gray. And this is the chi-square, and looking at the observed and expected, I got the chi value, and using the chi table, and I got a 15 to 20% probability. And my tri-hybrid cross, I crossed a purple female with a green male, and I got a 25% for the colors blue, red, and purple, and a 12.5 for green and gray. For my tri-hybrid cross, looking at the observed and the expected, I got a chi value of 9.614, and my probability, or my p-value, was 5 to 4%. And so my original hypothesis was if the genotype of the mono, di, and trihybrid zebrafish cross are known, then the phenotype of the progeny can be predicted. And it was supported, my hypothesis was supported for the mono di, but not the tri. And so looking at the monohybrid, I got the 20 to 30% probability that the differences between these numbers were due to chance and not real differences. And for the dihybrid, it was a 15 to 20% probability that the differences were due to chance and not real differences. And again, looking at the trihybrid cross, the probability was a 5 to 4 probability that the differences were due to chance and not real differences. And the 5 to 4 probability is showing that my hypothesis was not supported. And so I came to the conclusion that Punnett squares can be used to predict the outcome of the mono and dihybrid zebrafish cross, the phenotype and number of progeny, but not for the trihybrid cross. Last year, in 2017, I found that the green transgenic glowfish consume more oxygen per mass of fish, showing that they are less fit than all other fish. And so, as you are looking at the green, it may be impactful on the fitness and survival rate of the fish, and as well as the outcome of the cross. I would like to thank Dr. Jennifer Leung at the UMG Bio Department for the use of her lab and guidance throughout the study, Dr. Cynthia Wells, my science mentor, for her support and guidance throughout the study, and Francis Slater, a senior at Cloquet High School, who's been studying zebrafish for 5 years, and her guidance through my study. And these are my references. Thank you.